Hello and welcome to another episode of TWiML Talk, the podcast where I interview interesting people doing interesting things in machine learning and artificial intelligence. I'm your host, Sam Charrington. In today's episode, we're joined by Jay Newby, Assistant Professor in the Department of Mathematical and Statistical Sciences at the University of Alberta. Jay joins us to discuss his work applying deep learning to biology, including his paper, Deep Neural Networks Automate Detection for Tracking of Submicron Scale Particles in 2D and 3D. In our conversation, Jay gives us an overview of particle tracking and a look at how he combines neural networks with physics-based particle filter models. We also touch on some of the unique challenges to working at the micron level in biology, how we evaluated the success of his experiments, and the next steps for his research. A couple of quick meetup-related updates for you. Our next TWiML online meetup is coming up on September 19th and will feature David Clement presenting the paper Deep Mimic, Example-Guided Deep Reinforcement Learning of Physics-Based Character Skills. This should be a fun one, and I encourage you to join us. Next up, our second session of the Fast.ai study group is starting soon. If you've got some coding experience and would like to learn state-of-the-art deep learning, this is a great way to do it. To join either of these programs, please sign up for the meetup at twimlai.com/meetup. All right, on to the show. All right, everyone, I am on the line with Jay Newby. Jay is an assistant professor in the Department of Mathematical and Statistical Sciences at the University of Alberta. Jay, welcome to This Week in Machine Learning and AI.
Thanks, Sam. I'm happy to be here.
Awesome. Well, let's get started by having you tell us a little bit about your background and your research focus.
Thanks. My background is applied math, specifically modeling biological phenomena. I study small things that move in small spaces. So small things can be pathogens like virus or bacteria. They can be biomolecules. For the most part, I study things that are moving around inside of cells, but I also study extracellular spaces too. So these tend to be spaces that are on the scale of a micron, and the objects can be anywhere from a few nanometers on up to several microns. So one of the hallmarks of motion at this scale is that objects, when they're in a fluid, like inside of a cell, they move randomly. And so my area of mathematical focus is stochastic processes, and these are dynamical systems that have random elements. So something that's changing randomly through time. And that describes the motion of molecules and small objects in living systems. My PhD thesis was on modeling molecular motor transport. Molecular motors are molecular machines that move things literally. They burn energy, fuel to move objects, cellular resources, large distances. And we studied that in neurons. Neurons are unique among cells because they have to cast this very wide net. They have to cover so much space to construct neural networks. And that requires actively moving cellular resources around. And we studied the role of molecular motor transport in learning and memory. And that's, I guess, what led me to study particle tracking.
And so when you say particle tracking, what specifically are you referring to? Or is there a specific type of particle that you're interested in this research?
So particle tracking really refers to taking images with a microscope of these small objects in small spaces, right? So this can be, the cells to virus to many different things. So we take videos of these objects moving, and then we localize the center of the object through time and track its motion. And from that, we can learn many things about hidden factors, things we can't see about, for instance, the fluid of a cell or the of a mucosal barrier, Mucosal barriers, by the way, they defend us against infections and among many other things. So we can learn things about stuff we can't see by analyzing the random motion of these particles. But we also learn things about the objects themselves. So for instance, we might be interested in how bacteria move around in mucosal barriers. Because as we know, there's this very rich ecosystem that we're becoming aware of that live in our GI tract, our lungs, and they play this very symbiotic role in our physiology. So really, this task of particle tracking has many different applications in biology.
And your research is using images and neural networks to do this tracking. Can you talk a little bit about the origins of that work? Have you been using images and neural nets for a while?
Sure. So my background, as I said, isn't in image analysis or machine learning. When I started a postdoc at UNC Chapel Hill with Greg Forrest, I was working on many different projects, and at the root of all of these projects, the data source, it was all particle tracking data. So we would take, this particle tracking, which consisted of what we call position time series. So, X, Y, Z, T basically for a list of different particles. And then we would apply stochastic modeling to try and learn things. So it became apparent to me that I needed to really study how this was being done, because it's this mixture of software-assisted tools based on traditional image analysis and really, really heavy human interaction. So it's a very labor-intensive process. So as I learned more about how particle tracking software worked, I started to think about ways to automate it. And that's what led me down the road to learning about convolutional neural networks. It's kind of a weird road. My PhD advisor had done a lot of research on modeling visual cortex, specifically explaining how visual hallucination patterns form through various perturbations like drugs or injury, and how those form in the visual cortex. So I was sort of like vaguely aware of how neural networks in the visual system worked and are organized. And so I found it was one of the most amazing things reading how these things were actually trained, and that these feature the pattern, these sequential layered pattern matching was spontaneously emerging from the training procedure. So I tried to apply that technology to the particle tracking task, because really what is very time consuming about particle tracking is a very mundane task that humans are really good at doing over short periods of time. And that is just staring at a screen at a fixed image and pointing out bright blobs. I mean, particle
sounds very glamorous.
I know it's a very tedious job, and it's usually left to students and lab technicians, and it's very time consuming. The patterns, the way these show up in a microscope image. Well, I should back up. So first of all, say for virus typically are, around 10 nanometers to several hundred nanometers in diameter. And almost all of them are below what we call the diffraction limit, meaning that we can't resolve them with ordinary light microscopes. So they show up as blurry blobs, sometimes with these diffraction, like these wavy patterns. So picture dropping a pebble in a pond and then freezing that, the ripples, and then putting them around the bright blob. And that's kind of what it looks like. It's called an airy disk, right? And so you get these superimposed particles with little waves around them, and then you have to find the center of those patterns, right, where the particle center is. And when the wave-like patterns intersect, sometimes that can really throw off a particle tracking software and generate lots of false positives. So the patterns are relatively simple. However, microscope images do present some unique challenges. So first of all, we're talking about biological fluorophores that emit light that we are trying to detect with the microscope. And they emit very small amounts of light. We have to have very sensitive cameras that detect, and they're getting very, very good. They can detect upwards of 98% of the photons that they're being exposed to. But that comes with a price, and that is very low, what we call signal-to-noise ratio. That's a common term. I think that people understand. So these images are very noisy. So the patterns might be simple, but the conditions are very harsh.
So I did an interview not too long ago with a guy named David Van Valen, who's doing something very similar at the cellular level. He was using, I think, kind of the annotation software that he was using before he started looking at applying CNNs was something called Atlas Toolkit. Have you heard of that, or do you use something similar?
I've heard of the Atlas Toolkit. I haven't used that. I'm aware of David's work. I tried before encountering his paper to do the very similar thing, and that is cell segmentation. It's a very similar idea to track cells as it is to track particles. It's very difficult to do the segmentation part of that, to distinguish one cell from another as individual entities when they're very close together. That's a big challenge. So I've just started my group here at the University of Alberta, and that's one of the directions that I would like to go. There are many, many people I've talked to that are very interested in being able to do cell tracking. So there's a lot of opportunity, I think. And I think this is a great playground for people more broadly interested in object tracking because the patterns are very simple. So it's a great way to do a lot of experimentation and to also have an impact on the scientific community.
And so with your work with at the particle level, I imagine that segmentation comes into play in addition to, it sounds like you're trying to maybe relate some particles between frames and maybe determine motion vectors and things like that from one frame to the next?
That's right. Yes. We try to do that in a way that is as robust and widely applicable as we can. But we also have the option to bring in physical models. And this is where my background as stochastic processes in stochastic processes comes in. It's hard to write down a really physics-based model for the motion of a person, for example.
Right.
But we can write down very good models for the motion of a molecule that's undergoing Brownian motion. The classic example of a stochastic process, right? It's the drunkard's walk. You imagine that at every time point, you roll the dice, the proverbial dice, to figure out where you're going to step in the next instant and how far you're going to step. And you assume that you don't have a bias or a preference to go in any particular direction, and so you just wander about aimlessly. And that is what we see particles doing in these videos. So it's a very concrete and direct and valuable mathematical model. But we can do this for even more exotic, say, for example, molecular motors, which sort of processively move in a particular direction, and other things as well. So the tracking through time has its own unique challenges, but of course the main principles are very similar to tracking of sort of the macro human scale objects as well.
One of the recurring themes that I've explored in the podcast is this idea of combining neural networks, deep learning, with physics-based models, and it can be challenging. Can you talk a little bit about how you went about that?
Well, right now, we can apply methods separately. So we do like at first we'll run the CNN and do object recognition, find the centroid of all the particles, and then we kind of have this separate tracking step.
Okay.
Now, the tracking step can also employ machine learning methods like hidden Markov models, Bayesian networks, basically particle filters. And that's an unfortunate name conflict, right? The particle and particle filters is not the same as particles and particle tracking, but those ideas are the same. So those physical models port directly, these stochastic models of the motion port directly into those methods. Then everything is an optimization. And we can even infer things that are hidden, things that we don't see, such as, the hidden state of a bacteria, which uses a flagella like a motor to move around actively, and they spread out much more quickly this way, and so we can determine if a bacteria, say, is using its flagella right in a certain way, or if it's just passively sitting around. And it makes a very powerful tool. And what's great about it is all the physical assumptions, right? These are very important when you're writing a scientific paper. You want to explicitly say all the assumptions that you make in your analysis pipeline. And when you use a method like this, they're all in the model, all in this physical model, right? And then everything is an optimization, just using standard Bayesian statistical tools. Everything is an optimization, finding the best parameters, the best fit for that model.
You mentioned particle filters. Can you explain what those are?
Particle filters, they're basically a way of taking observations and inferring hidden factors that are so you might assume that you have, I think the classic example is a GPS detector in a truck. And so you would like to be able to get an inference of the truck's position based on the noisy GPS recordings. So you have some sort of model that relates the GPS recording to the truck's actual position, but also where the truck has been in the past influences the inference of where it is in the next sort of like time step. And so you have an observation model and a motion model, and you build these all in together. So from the neural network perspective, I think the closest analogy maybe is the recurrent neural networks, which use kind of like information about the past, the most recent past, to make inferences about the future.
And so, given your familiarity with David's work and some of the audience's familiarity with that work, I'm wondering if there are specific, unique challenges that you encounter here working at the micron level.
Yeah, there are many challenges. One being that microscopy sort of developed alongside, in the computer age, regular photography, I guess you could say, or video compression. So all the formats and everything there's a million of them, and they're all different. So it's difficult to work with the data. Most of the time, everything is uncompressed, so the data sets can be quite large. And add to that the difficulties of imaging biological entities that are very dim, and they're emitting very low levels of light. And so you also have to deal with very low signal to noise ratios. So we actually also do 3D video microscopy, and that is another unique challenge here. And so how do you apply this convolutional neural networks to 3D videos and to also extract useful information there. So 3D video sets, of course, are much larger than their 2D counterparts. So we have to actually build up special tools to be able to work with those.
And in the case of 3D, it's using stereo microscopes. Is that where the 3D models come from, or are they generated in a different way?
So one of the projects that we're really excited about now is doing particle tracking inside living cells. And so we do 3D particle, we collect 3D videos. So we try to image the entire cytoplasm of this cell. And the cytoplasm is just the fluid on the inside of the cell. So the way this works is that the microscope has a stage, what's called a piezoelectric stage, which is it just has a motor that can move really fast and very accurate small motion movements. And so it moves that stage up and down, and then the camera just takes the images sequentially. So you kind of one layer at a time, you build up a 3D volume, and then it's like a typewriter. You collect one volume, and then you restart to the beginning again and collect the next volume. So, and in that way you get this time sequence of volumes that comprise a three-dimensional video.
One of the things that struck me in looking at some of the images of these particles is that they're very dense. And so there's a lot of occlusion, there's a lot of overlapping. Did you have to do anything special to deal with that?
Well, the answer is yes and no. So that is a very difficult problem that you identified. So not only that, but the particles are appearing and disappearing. They're overlapping, occluding, but then they leave. They're too dim. They basically leave what we call the focal depth that we're actually imaging. And then of course, since they're moving randomly, they can come back in. So we have to be able to track them only while they're there and decide when they're gone. And the way the neural network does this, it automatically does a couple of things that allows us to at least have a first start, right? That doesn't generate any. It's not perfect. So, for example, when two particles overlap each other in 2D microscopy, what the neural network will do is just localize a single particle in that position. Leading up to that, or just after they overlap, one of the particles is typically dimmer or smaller, and the neural network will pick up only on the stronger signal. So what typically happens is we just stop tracking an object, right, once two particles get close enough. And so it does chop short that particular particle track, and we might pick it up again later. But this is preferable to making errors and mistakes. So we can actually do things to correct for this on the linking stage, where we assemble these centroids into tracks. And there's been probably 10 to 20 years of work on this problem, actually. Some really great methods out there for this. So we're in the process of kind of building up this toolbox to correct for these little things that we see. And one of the other things I mentioned was that particles appear and disappear. So the neural network always outputs a confidence, right? It's not just a yes or no answer. It tells you how confident it is about a classification. We do, foreground versus background, so it's just a binary classification. But when particles are dimmer and they're about to leave the field of focus, that confidence goes down. And so we can actually take that information into account when we're deciding how to link a particle in one frame with an observation in one frame with an observation in the next frame.
And so you're using CNNs to do this. Does that mean that someone went through and manually annotated some number of frames in order to give you training data?
Great question. So yes, somebody did do that. That somebody is, well, one of three people was me, and that was not the funnest thing I've ever done. Um, but the funny thing is, we actually did end up using the hand annotated data. So in the lead up to this, as I was learning and frankly learning about this technology, CNNs, how they work, experimenting around with them, I just built up synthetic data, just simulated the way that these videos looked. And over time, that built up to be good enough that when we trained the neural network with the synthetic data, I was never able to get anywhere close to the accuracy when I replaced that synthetic data with manually segmented data. So I don't know why. I don't know what the reason for that is. I think my guess is that it's because the ground truth was so absolute in the synthetic data set, and we were able to randomize the appearance and get the appearance accurate enough that we were able to get a robust neural network out of it. This is basically impossible, right, to do with human scale images. How do you synthetically create an image of a cat? I mean, that's a very hard problem in of itself. But for the scale we were working on, because these objects tend to be below the diffraction limit, like I explained, they're these blurry, wavy patterns. They're relatively simple to create synthetically.
Huh. Oh, that's really interesting. And so what types of you mentioned that you applied presumably some kind of noise or filters to augment your data, or at least tune it to get as realistic as possible. What can you elaborate on some of those things that you did?
Yes, I randomized everything. So every sort of parameter I could think of, like how the particle size, details about the shape. I put random background patterns, random amounts of background noise. I tried to shotgun as wide a field of conditions as I possibly could. So that we could in turn, use this on the idea is to automate this. And what we found in extensively testing it for over about the last 2 years now, on data from dozens of labs, is that it is surprisingly robust to different conditions. The conditions that were outside of what we ever trained it on. For example,
and just a point of clarification, are you saying that the training data that you developed is robust and other people could build models against it, or the models that you built are robust and they work with other people's images?
The second, right?
Okay.
We've seen that the tracker is capable of accurately tracking conditions that we've received from labs that were beyond what we actually included in the training data, the synthetic training data. One example is tracking salmonella. These are about 1 to 2 microns in size, so they're above the diffraction limit, barely, and which means that we can actually resolve the shape, and they're rod shaped. So we trained everything to recognize like these rotationally symmetric, blurry shapes with the waves, and so as we get images that have perturbations from that rotationally symmetric pattern, we still get recognition. So we can track rod shaped. We've tracked comet shaped objects, lots of different kinds of backgrounds. So we never trained it to ignore large bright spots. These show up all the time though in experimental videos. But the neural network tracker evades those, it ignores those. So it's been surprisingly robust. And that's really the key here of what we see this technology is enabling for us is automation.
Do you think that the advantage of your synthetic data over the manual, granted you said you're not really sure why it worked the way it did, but did you have just a lot more of it? Could it be explained by volume alone, do you think, or did you compare comparable training data set sizes, synthetic versus real?
I don't think it was the size of the data set. We had a fairly large set of hand segmented data. It gets so, and this is part of the problem, it's very subjective. There are certain really bright particles that show up and there's no ambiguity. There's no mistaking what they are. But you start getting, so particularly when the particles get dimmer, they're about to leave the field of focus, and reappear. It's a very subjective call whether or not to label them as particles or not. So I think that probably has more to do with it. The synthetic set is very consistent. So I tried layering in the hand segmented data with the synthetic data with varying proportions, and the performance always went down sharply with the amount of the hand segmented data that we used for training. I can't give a definitive reason why that's true.
It's very surprising relative to the way we usually think about this manually produced ground truth data. And also the degree to which these CNN models are, they can be very sensitive to undiscernible characteristics of your actual data. And so the simulated data that you think looks great, and close, totally confounds these models because it's missing some, undiscernible or very subtle nuance that is in the real data. So it's a very interesting result.
I completely agree. I was very surprised when I learned about this technology, everything that I was learning, that real data was absolutely essential. Just turned out surprisingly not to be the case here.
And so you developed the model. How did you combine the model with the tracking element?
So how do I combine the output of the convolutional neural net with
the path linking piece?
Yes, right. So right now they're actually quite separated. So the first stage is processing with the neural net, and then we segment and what we call localize, which means just estimate the centroid of the spots, and that's all basically done with the output of the neural network. And then we take this disorganized or unsequenced collection of particle locations at each frame, and then we link them together. And that is done. The most robust and most widely applicable method is just to use something called the Monk-Res algorithm, which just says, I'm going to take two sets of objects and find the best match between all of those objects that sort of minimizes some cost. In this case, distance. So that's a classic tool in object tracking, and we use an adaptive method. Again, we also have to decide when to stop tracking or when to start tracking a new object that's appeared, and we do that with the confidence from the neural network output. But there are a lot of possibilities, I think, that are very exciting in machine learning for unifying both of these parts together, and really taking the linking side, maybe using a stochastic model that's physically based or not, but using machine learning and training end to end, not just end to end in the CNN, but end to end between the object recognition and the object tracking side.
How did you evaluate the ultimate performance of the experiment? What were the key metrics that you were tracking?
So we looked primarily at the success of object recognition in our first paper, and so there it was an assessment of basically false positives, false negatives, right? But also how accurately we detected the center of that particle, the centroid, which is important in a lot of applications. So we did a combination of synthetic videos that randomized across a lot of different conditions, so that we could sort of assess the robustness. And this was mostly to compare it to existing software that's already out there, to emphasize the software that the currently existing software really requires manually tuning parameters on a per video basis. So we wanted to emphasize the automation part. So that's on synthetic training data. But we also, of course, tested it on experimental videos as well. And those were generated in Sam Lai's lab, where we were tracking synthetic nanoparticles, virus, bacteria, and then assessing again the object accuracy. Since then, we have a number of collaborations around the last 2 years where we've applied this technique, and so we've, I guess, field tested it pretty extensively as well.
You mentioned that when you incorporated the real data, your performance dropped significantly. How would you characterize the performance differences between just applying the model to synthetic data versus real data?
It's a great question. So, of course, the synthetic videos that we used to test it were minor alterations of the synthetic videos that we used to train it. So, of course, it's going to perform very well on those, even though all the conditions are sort of randomized. The real data, there have been some surprises, little things where the neural network has failed, and so that's given us the opportunity to tweak things over time and improve things. For example, we noticed that sometimes there is this mixture of intensities, pixel intensities, so how bright the objects show up. So there might be some particles of type A and type B, for example, some that are very dim and some that are very, very bright. And the neural network would only track the bright ones. So that's something we had to go back and we built that into our synthetic video generation. So the workflow has been that we notice something that the neural network doesn't quite do correctly, and then we go back and build that into the synthetic video generation and retrain. And that's worked really well for us.
What are some of the next steps for your research group and in this research direction in general?
Well, I mentioned before that we're very excited about doing particle tracking inside living cells. This gives us a spatiotemporal measurement of the entire cytoplasm of a cell. So we can measure things like crowding, confinement, viscosity. You have to remember that all of the chemical reactions, a chemical reaction is just molecule A, molecule B coming together, and they come together by random motion, by Brownian motion usually. So they're moving through this fluid, and all of these things like crowding and confinement and viscosity, they influence the chemical reactions, all of them that are happening inside the cell. So everything that's going on inside physiologically of a cell it depends on this. And so this gives us a quantitative measurement, a window into that, into these conditions. Like I mentioned before, we're doing 3D, we're collecting 3D videos for this. And one of the real challenges there has been to deal with these large data sets. So a 2D experiment usually generates around 1 to 50 gigabytes of video data, where a 3D experiment is going to be roughly 10 to 20 times larger than that. So we're easily, a single experiment can generate, terabyte or more of data. So we needed, and 3D videos themselves are very large files. So we needed a way of implementing the neural network, right, that could handle these large videos. And we did this with cloud computing resources, we've been using Google Cloud, which has been extremely generous in supporting our commercial and our research projects. And we've been using Apache Beam to basically implement a MapReduce style processing pipeline to break up these videos and process them one piece at a time. The great thing about Apache Beam and Google's data flow is that it dynamically manages the hardware. So if I have a small set, maybe I only need 20 CPUs. If I have a terabyte, maybe I need 1,000 CPUs over hours, and it dynamically instantiates those virtual machines and then shuts them down when they're no longer needed. We're also very interested in delivering this as a commercial web app that people can use. We want to empower biologists working in labs that typically don't have a technical computer science background with programming skills. So we needed to build something that's very easy to use, platform independent, could handle large sets of data, and so a cloud-based web app seemed like the best option for us.
Early on in the discussion, you mentioned a couple of application areas. One was looking at neurons, and the other was looking at some of these micromotors or cellular motors. Did you learn anything through the development of the particle tracking system about the systems that you ultimately care about?
Absolutely. That's ultimately what we're most excited about doing, is learning new things biologically speaking. So we have applied this to tracking these small fluorescent molecules. We call them GEMs. They're actually synthesized by the cell. They're about 30 nanometers in diameter, and we're looking at how cells basically program the cytoplasm, the fluid that everything moves around inside, in these fungal cells. They're called Ashbia gossypi. This is in collaboration with Amy Gladfelter's lab at UNC Chapel Hill. And what's special about these cells is that lots of different nuclei, multiple nuclei, share the same cytoplasmic space. They're called syncytia. Our typical picture of a cell is one nucleus with DNA, right? But syncytia actually are not that uncommon. We have them in our own bodies, muscle cells, for example. So it's an interesting question to understand how multiple nuclei coexist. They all have to undergo division like a cell would. There's a cell cycle, and all those cell cycles conflict with each other if the nuclei are too close. So we are using particle tracking to try and measure how the cytoplasm is regulated, programmed in order to isolate these nuclei, or maybe they cooperate, maybe they compete. There's a lot of questions that we are interested in answering. So this is an ongoing project right now. And we also study mucosal immunology. So we are interested in how virus and bacteria can penetrate a mucosal barrier, which could potentially lead to an infection. So we discovered, this is with Sam Lai's lab at UNC Chapel Hill in the School of Pharmacy. We discovered that these mucosal barriers actually trap virus by using antibodies, which is a potentially therapeutically exploitable avenue of protecting against infection.
Do you already have some areas of further application of machine learning to your work?
Absolutely. So one of the things I would really like to do is move to more the human scale tracking world, and to keep going with object tracking applied to scientific problems, but to track, say, fish or fruit flies or worms in controlled laboratory settings where you want three-dimensional information, possibly from 2 or 3 different observation points. And there, I would like to be able to use mobile hardware to process this in real time, to process and track images in real time.
Oh, really interesting. Well, Jay, thanks so much for taking the time to chat with us about what you're working on. I appreciate you walking us through it.
No problem. Thanks for having me.
All right, everyone, that's our show for today. For more information on Jay or any of the topics covered in this episode, visit twimlai.com/talk/179. If you're a fan of the podcast, we'd like to encourage you to head to your Apple or Google Podcast app and leave us a 5-star rating and review. Your reviews help inspire us to create more and better content, and they help new listeners find the show. As always, thanks so much for listening, and catch you next time.